Welcome to the Differential Gene Expression module, where we're going to learn about transcription factors. We will begin with learning about the properties of transcription factors, as well as the different domains that are normally present in transcription factors. We will learn how transcription factors regulate gene expression, as well as different types of transcription factors, with the focus on pioneer transcription factors and master regulatory transcription factors. Transcription factors are proteins that are able to bind to DNA and regulate gene expression. Now, transcription factors are able to activate transcription by recruiting histone modification enzymes, and another way they can activate transcription is also by stabilizing the pre-initiation complex. Transcription factors can be grouped into different families that are based on their DNA binding domains. In this table, we can look at different families, which are the homeodomain family, the basic helix-loop helix family of proteins. We also have the basic leucine zipper family of proteins, as well as the zinc finger family of proteins. When we look at the transcription factors, they all have certain domains. The first one is the DNA binding domain that will recognize a particular DNA sequence in the enhancer. The domains or motifs that allow DNA binding to occur in the transcription factor are the homeodomains, the zinc finger domains, the leucine zipper, helix-loop helix, and helix-turn helix motifs. Another domain that is commonly present in transcription factors are the transactivating domain. This domain can activate or suppress the transcription of the gene whose promoter or enhancer it has bound. The domain also enables binding with general transcription factors and/or chromatin modifying enzymes. A third domain that is commonly observed in transcription factors are the protein-protein interaction domains. They allow the transcription factor's activity to be modulated by other proteins, and they normally enable dimer formation in some proteins. We're now going to look at another DNA sequence called as an insulator sequence. Now, these DNA sequences limit the range over which an enhancer can activate a promoter. They can establish the boundaries of gene expressions, and thus the insulators help ensure that a promoter does not get activated by another gene's enhancers. Now, when we look at the figure below, let us look at the top one, which shows that there is this enhancer E1 that can activate the transcription of genes to the left as well as to the right. Now, it is important to note that the location of an enhancer can be upstream of a gene, but it can also be downstream of a gene, and hence its location is not fixed the way the location of a promoter is fixed. Now, let's look at the second scenario in the figure where right after the enhancer shown as E1, there is an insulator sequence. Now, in that case, the enhancer is able to activate the transcription of the genes to its left, but the presence of the insulator sequence does not allow the enhancer to activate the transcription of the gene to its right. And thus, the insulator ensures that a promoter is basically not activated. Now, transcription factors can bind to insulator sequences to change the conformation of the chromatin and separate the enhancer from the promoter, and that is one of the mechanisms by which insulators do not allow transcription to occur. One of the well-studied proteins that can bind to insulator sequences is a transcription factor called CTCF, which binds to the insulators and interacts with cohesion, which can change the conformation of chromatin. If we look at the figure below, let's look at the middle panel first. In the middle panel, that oval-shaped structure is the promoter of a gene. And when we look at the enhancer, we can see that it is quite far away from the gene. The cylindrical structure between the promoter and the enhancer is the insulator sequence. Now, when the gene is activated, that normally happens when there are no proteins bound to the insulator, but there are proteins or transcription factors bound to the enhancer and the promoter, and this causes the transcription of the gene. Now let's look at the rightmost one, where we have a transcription factor, CTCF for example, that has bound to the insulator sequence. The binding of CTCF to the insulator sequence changes the conformation of the chromatin such that the enhancers are now far away from the promoter, and thereby even if you have proteins bound to the enhancer, they have no way of contacting the promoter and promoting transcription of the gene. Thus, the insulator is able to prevent transcription of genes. We're now going to look at a class of transcription factors called pioneer transcription factors. These are transcription factors that can penetrate repressed chromatin like heterochromatin and bind to their enhancer DNA sequences. So basically, if we have DNA that is tightly packed, but we want the genes that are present in that heterochromatin to be expressed, we have to have a way to be able to loosen up the chromatin. And in that case, the pioneer transcription factors are able to go and bind to enhancer sequences present in tightly packed chromatin. They aid in opening the chromatin to now allow other transcription factors to bind to the promoter sequences. As shown in the figure, here we can actually see that on the top figure, the pioneer transcription factors are binding to sequences on chromatin that is tightly packed and are able to recruit proteins that loosen up the chromatin. These pioneer transcription factors play important functions in establishing certain cell lineages during development of organisms. For example, FOXA1 is a pioneer transcription factor that is required for liver cell formation, while PAX7 is required for muscle formation. Another class of transcription factors that we're going to look at are the master regulatory transcription factors. These are transcription factors that are able to control cell differentiation. The requirements to be a master regulator transcription factor are that they must be expressed when the specification of a cell type begins. They must regulate the expression of genes specific to that cell type, and they must be able to redirect a different cell's fate to this cell type. Basically, the whole concept of a master regulatory transcription factor began when scientists found that if the nucleus of a frog egg was replaced with the nucleus from a terminally differentiated cell, the egg developed into a normal frog. So there was something in the cytoplasm that helped the nucleus to go ahead and enable the whole process of development to get a frog. And so what components in the cytoplasm aided this process? And that is how master regulatory transcription factors came to be known. Nowadays, some master regulatory transcription factors have been identified that can actually change a cell's fate. Let us look at an example where we're going to take cells that are differentiated mouse fibroblasts. So the type of the cell is that of a fibroblast, which means it has already undergone differentiation. When scientists added 4 transcription factors to these mouse fibroblasts, and they were the OCT34, c-MYC, SOX2, and KLF4. After adding or introducing these 4 transcription factors to the differentiated mouse fibroblasts, what they found was that the resulting cells actually changed their differentiation process and basically became undifferentiated cells that are basically stem cells. So they were called as induced pluripotent stem cells because these cells now had the ability to become different lineage cells like mesoderm lineages, ectoderm lineages, or endoderm lineages. So in this case, the cells started out as mouse fibroblasts, but by just adding 4 transcription factors, we were able to change the cells to become pluripotent stem cells that could now be coaxed to differentiate into other types of cells. Thus, OCT34, c-MYC, SOX2, and KLF4 can be considered as master regulatory transcription factors. These master regulatory transcription factors are becoming more and more popular to reprogram cells. For an example, in the case of a mouse model where the mouse was diabetic and the pancreas were unable to make insulin anymore, scientists went ahead and introduced 3 genes into the pancreas of this diabetic mouse. The 3 genes were PDX1, NGN3, and MAF-A. After the injection of the vector that had these 3 genes so that it could be introduced into the pancreas cells, the scientists looked at the expression of insulin in these pancreas cells. Now remember, the mouse is the diabetic mouse, and so it's diabetic because its pancreas could no longer synthesize insulin. However, by adding these 3 transcription factors, what the scientists found was that they were able to make the pancreas cells change their programming so that now they were actually able to make insulin, and now the mouse is no longer diabetic. There are a lot of experiments going on to try to treat diseases in this fashion by expressing transcription factors and reprogramming cells so that disease cells can now be functional and we can treat diseases in this manner. The figure below basically tells us how these 3 transcription factors are involved in the formation of different types of cells in the pancreas. The beta cells of the pancreas are the ones that actually make insulin, and that is why the scientists had used those 3 transcription factors to convert or redirect the pancreas cells to become beta cells so that they could make insulin.
With this, we come to the end of our talk where we learned about different aspects of transcription factors.